Swayam Prabha. Digital India. Educated India.
Welcome to module 3 of my course Genome Editing and Engineering. In this module we have been discussing about the recombination process. Today we are going to discuss about the site specific recombination. Site specific recombination encompasses a group of processes in which DNA molecules are rearranged by breaking and rejoining the strands at specific points. So it involves excision, insertion or integration and inversion. We will be discussing how this happens in the course of our lecture. Let us have a understanding of the homologous recombination versus site specific recombination. In the last lecture we discussed about the HR, which utilizes long stretches of homology between partner DNA molecules for double strand break repairs. This process involves a large number of proteins with distinct biochemical activities which cooperate to carry out homologous recombination. In contrast, site-specific recombination targets relatively short DNA sites with well-defined sequences and usually a single protein or a pair of proteins is or are involved in carrying out the catalytic steps of site-specific recombination. Sometimes the site-specific recombinases are aided by one or more accessory proteins in synapsing the DNA partners and assembling the chemically competent recombination complex. Due to its simplicity, these systems has been utilized for bringing about targeted genetic alterations within genomes. Site-specific recombination involves two short DNA sequences or sites, which may be within the same molecule or in different molecules. A specialized recombinase enzyme recognizes the sites and then rearrange the DNA such that the left end of one side becomes joined to the right end of the other side and vice versa. In classical site specific recombination, the strand breaking and rejoining reactions are catalyzed by a recombinase and are highly conservative. It do not involve any DNA synthesis or degradation or any enzyme cofactors. Briefly, site-specific recombination are a set of specialized processes that involve reciprocal exchange between defined DNA sites, and in general it requires the following. Number one, it requires two DNA partners. It requires a specialized recombinase protein that is responsible for recognizing the sites and for breaking and rejoining the DNA. And thirdly, a mechanism that involves DNA breakage and reunion with conservation of the phosphodiester bond energy. Some examples of SSR or site specific recombination which meet the above criterion are the integration of bacteriophage lambda into the E. coli chromosome, the resolution of co-integrates derived from transposition of TN3 related transposons, and a DNA inversions responsible for flagellar phase variation in Salmonella. This narrow requirement, however, excludes several other specialized recombination processes that have been on occasion been described as site specific recombination. These will be discussed as and when necessary. Some of these are VDJ recombination catalyzed by the RAG1-2 proteins during the development of the immune system or immune reactions. Most DNA transposition events, even when a site specific target is used, including integration of retroviral cDNAs and the homing of mobile introns. What is the outcome of the process of site specific recombination? You can see in this figure there is a candidate gene and then there is a sequence of interest, and these are flanked by say the candidate gene is flanked by some genetic sequence 1 and 2, the target sequence is flanked by some sequence 3 and 4, and if this candidate gene is to be integrated, you can see there is a change in the flanking genes, the candidate gene carrying along with it the flanking gene 1 to the sequence site, which is now having on the right side the flanking gene 4. So, this is just the schematics of the outcome of the site specific recombination. So, this however depends on the initial arrangement of the parental recombination sites and gives rise to either insertion, integration or excision. Sometimes instead of this insertion there may be excision and there may also be inversion. Now, this integration results from recombination between sites on separate DNA molecules, provided that at least one of the parental chromosome is circular and occur with a uniquely defined orientation. For sites located on the same chromosome, the outcome is determined by their relative orientation. Site specific recombination may also lead to excision of genes. So, there is a candidate gene over here, and now as excision has happened, you can see the gene is no longer in the scene and it has disappeared, and in the case of inversion you can see here the gene has been inverted. For sites located on the same chromosome, the outcome is determined by the relative orientation. Excision results from recombination between sites in a head to tail orientation, while inversion results from exchange between inverted head to head sites. So, this is one of the important papers which was published in the PNS, and Gates and Cox reported the FLP recombinase and its enzymatic activity. Enzymes which catalyze site specific recombination between two specific DNA sequences and mediate DNA integration, excision, resolution or inversion are called as site specific recombinases. These site specific recombinases, which play a pivotal role in the life cycles of many microorganisms, are classified into two types: tyrosine type or serine type recombinases, and this classification is based on whether a tyrosine or serine residue mediates the catalysis. They further divided or subdivided into two subfamilies, each based on the following criterion. The tyrosine type recombinases are subdivided by the direction of the recombination or recombination directionality, while the serine type recombinases are subdivided by the size of the protein. The tyrosine type recombinases catalyzes the formation of a Holiday junction intermediate and resolution. The catalytic mechanism of serine type recombinases includes the 180-degree rotation and rejoining of cleaved substrate DNA. And you can see here the eponymous Holiday junction, a model for DNA recombination intermediate proposed by Dr. Robin Holiday. And this is the simplified diagram where 4 strands of DNA participate, and this is the cartoon diagram of the DNA double strands. So, briefly we have up till now discussed about the different type of classification of the site specific recombinases. Broadly they fall into two types: tyrosine type and the serine type, and here the 3' phosphotyrosine linkage with cleaved DNA, and here you have the 3' phosphosericin linkage with cleaved DNA, and these tyrosine type are further subdivided into phase integrase, which may be unidirectional, and simple type, which may be bidirectional, whereas the serine types are divided depending on the size, like the resolvase or invertase, they may be small, and the phase integrases, which are very, very large. And there are other features which are important to these recombinases. For example, the phase integrase requires the host factors ISF and FIS, and these needs supercoiled DNA. These do not need the HF, and the serine types, particularly the small resolvases and integrases, also have some kind of directionality. They may be both uni and bidirectional, and the requirements of HF may vary. Heme and gene require the SF, FIS and HU. They also need supercoiled DNA and recombination sites in Cs, whereas the large serine type phase integrases do not need any HF host factors. All enzymes recombine to target sites which are either identical or distinct, whereas for A1 these sites have individual designations, FRT in case of FLP recombinases, LOXP for Cre recombinases. The terms ATTP and ATTB are attachment sites on the phage and bacterial part respectively, and these are valid in other cases as well. In case of A1, we have to deal with short, usual 34 base pair long sites consisting of two near identical 13 base pair arms flanking an 8 base pair spacer, the crossover region indicated by red line doublets over here. So, let us look into this table to understand this discussion. We have the types of various recombinases, tyrosine and serine. You have tyrosine recombination and tyrosine integrases, sericin resolvases, invertases or sericin integrases. And these are the sources, bacteria, phage and yeast, and you have the various members like Cre, Dre, FLP, KD. You have, for example, in the case of integrases, lambda integrases. In sericin resolvases, you have gamma delta, PAR-A, TN3 gene, and so on. And in certain cases, they require the accessory factors already discussed earlier, and some members do not. And these are recognition sites in phage and in the bacteria as discussed earlier. Let us first discuss about the features of tyrosine recombinases and what are the type of reactions they carry or the mechanisms involved. So, these tyrosine recombinases are found abundantly in prokaryotes, archaea and even eukaryotes, and in certain families of retroposons. Various structural studies of these tyrosine recombinases have shown that they share a catalytic domain with recognizable sequence motifs, and the fold of the entire domain is well conserved. Although some family members such as FIMB and FIMI contain only this domain, however, in most the catalytic domain is preceded by a variable N-terminal domain that helps binding to DNA. Some such as lambda integrases have a second N-terminal domain that binds different DNA sites. For instance, there are two tyrosine recombinases of unknown function whose catalytic domains are followed by putative molybdate binding domains. The catalytic domain is shared with at least two other classes of enzyme. Type 1b isomerases function as monomers to release supercoiling tension in DNA by cleaving and religating just one strand of DNA, but they do so through a similar 3-phosphotyrosine intermediate with an almost identical active site. The second one is the telomere resolvases or protelomerases. They maintain the covalently close hairpin ends of the linear replicants found in certain prokaryotes and viruses. Apart from the essential catalytic tyrosine residue, most tyrosine recombinases possess 7 conserved residues as listed here: ARG1, GLU-S1, LIS, histidine 2, ARG2, HIS and trypsin 3, tryptophan 3. These Roman numerals here 1, 2 and 3 correspond to the 3 catalytic motifs 1, 2 and 3. The tyrosine and lysine residues serve as a nucleophile and general acid catalyst respectively. The ARG1 and ARG2 residues neutralize the negative charge during the transition state and activate the sessyl phosphate by the catalytic tyrosine residue. The HIST, TRP3 and HIS2 and GLU-ASP1 residues stabilizes the transition state. Bacterial tyrosine type integrases contain a signature R1 H2 XS R2Y tetrad, where X can be any residue. Now, what is the mechanism by which this recombination happen? Tyrosine type Faz lambda integrase, which is a unidirectional tyrosine type recombinase. Let us study its mechanism first. Bacteriophages are classified into two groups: lytic and temperate phases. Once inside the host bacterial cells, lytic phases immediately propagate themselves by taking over the host cell machinery and release large number of phases by lysis of host cells. In contrast to these, the temperate phases enter either lytic or lysogenic life cycle after host cell invasion. In lysogenic cycle, the temperate phase genomes are integrated in the host bacterial chromosome at a specific site by the site specific recombination catalyzed by a phase encoded integrase during conversion to the prophase state. In the initial stage of the conversion to the lytic phase, a phase encoded excisionase catalyzes the excision of the prophase genome by cooperating with the phase integrase, and the original ATTP and ATTB sequences are regenerated via site specific recombination between ATTL and ATTR. So, the integrase and the excisionase carry out opposite functions. We have discussed about the ATTP and ATTB sequences earlier. So, you can see this location of these sequences over here. Now, in the case of integration, you need integrase, and excision would require the integrase as well as the excisionase, and these gives rise to the prophase. The activities of heterobifunctional tyrosine recombinases are frequently regulated at a structural level by excisionase, which alter the ability of the recombinase to assemble into higher order recombinogenic nucleoprotein structures. Integration of the lambda DNA. Lambda phase DNA contain an attachment sequence called ATTP as shown in this figure, and the bacterial DNA contain an attachment sequence called ATTB. Phase and bacterial DNA align at the O region of the attachment sequences. During integration, INT protein induces two double-strand breaks that are resolved. This lead to the integration of the fast DNA into the bacterial DNA. To switch between either lytic or lysogenic life cycle, the process as it happens is reversible. For excision of the prophase phase DNA from the bacterial DNA, INT protein and GIS protein is needed, as already discussed earlier. The integrated fast DNA oocyte is flanked with one side from the phage and one side from the bacteria. These are called the ATTL and ATTR sites. And we can see here the genome of the phage lambda, which has certain sequences corresponding to the head, tail, tail tip and tail fibers, and these are not expressed together. These are some early genes and some are late genes. Accordingly, they are known as the early operon or the late operon, and those which are located on the left side of the genome are the early left operon, and those which are expressed towards the end are the late operon. And here you can see the location of the site specific recombination and the ATT sites as well as the integrase and the GIS. So, these maps give us an idea that all these elements required for site specific recombination are under the early left operon. So, this is a magnified view of this diagram showing the various components here, as we have already discussed, as well as the elements or sequences which are involved in the general recombination. How does the phage lambda get integrated? So, or how does the excision of the phage lambda takes place? So, in the first case we can see the integration is happening, and in the second case the excision is occurring. So, tyrosine type phage integrase mediates site specific recombination. The ATT sites are recognized by phage integrase. Core type binding sites containing the binding sites for the integrase in ATTP and ATTB, and strand exchange occurs at a 7 base pair overlap region. ARM type binding sites in ATTB contain the binding sites for the integrase ISF and hexokinase. Let us now discuss about the bidirectional tyrosine type recombinases. You can see here a term causes recombination, and the C and the R, E, re are combined together to form CRE. So, CRE is short form for causes recombination, and this CRE is a recombination enzyme or recombinase enzyme from phase P1. Similarly, the FLP is a short form from flippage, and this is a recombinase from the yeast 2 micron plasmid cycle. Both of these catalyze reversible site specific recombination events between two short identical sequences, and these sequences are 34 base pairs LOXP sites for CRE and 48 base pairs FRT sites for FLP. Strand exchange occurs in the 8 base pair regions, spacer regions, and unlike others, these recombinations do not require host encoded accessory proteins, ARM type binding sites or specific DNA structures. how does the recombination by tyrosine type recombinases occur? So, in the first step, 4 recombinase monomers bind to the DNA substrates. So, this is a DNA substrate here, another DNA substrate here, and you can see here the monomers 1, 2, 3, 4, which together bind to the DNA substrates as shown in the picture. 2 of these monomers are inactive. These yellow ones, for example, are inactive, while 2 are active. Okay. You can notice here phosphotyrosine in position in the blue ovals here, which is appearing a little bit grayish. The active monomers cleave the first pair of DNA strands to form a 3J phosphotyrosyl intermediate and free 5J hydroxyl groups. Now, after these, there is a strand exchange which results in holiday junction intermediate, as you can see here, and this is followed by isomerization. Now, there is a conformational change in the next stage, or a rotation takes place, and the second pair of monomers which were earlier inactive now becomes active, and they carry out the second set of DNA cleavage. The second round of strand exchanges and ligations result in the recombinance. Now, you can see here a molecule, 2 different molecules, DNA molecules painted blue and red after a recombination process becomes a hybrid molecule as shown by these colors, having both blue strand and the red strand, and both the products are now recombinant products. Recombination by tyrosine type recombinases. So, here the 2 strands binds here, as already told to you, and then there is a strand exchange which forms the holiday junction, and this is the isomerization step which we could not see in the earlier pictures. And finally, the second cleavage occurs, and then finally a recombinant DNA products are produced. So, this is basically, as you can see, a 5-step procedure or 6-step procedure, including the isomerization step. Cre and FLP recombination systems bear several important mechanistic similarities to the integrative recombination system of Bacteriophage lambda and are members of the INT superfamily of site-specific recombinases. One major characteristic shared by recombination systems in this group includes the formation of 4-stranded holiday junction DNA intermediates. FLP is a site-specific recombinase encoded by the 2 micron circle of the yeast Saccharomyces cerevisiae. It is thought to be responsible for maintaining multiple copies of these extra chromosomal elements in vivo. Here, Cre recombination system of the fast P1 resolve tandem dimers of the P1 genome that form during replication, permitting the proper segregation of daughter fast genomes at cell division. Let us now discuss about the serine recombinases. The serine recombinases generally bind their individual crossover sites as dimers, but the strand exchange reaction itself occurs within a tetramer. Double strand breaks are introduced by the attack of yeast promoter's active site serine on a sessile phosphate group, leading to 2 nucleotide 3' overhangs. 2 subunits are then hypothesized to rotate 180° relative to each others, aligning the cleaved DNA ends with new partners. The religation reaction is chemically the reverse of the cleavage reaction, with the 3' hydroxyl groups attacking the phosphoserine linkages. You can see here the serine type recombination catalyzed reaction. So, you have 4 units and they are similarly binding 2 different DNA molecules, and then 2 of these are on the top and bottom, and they bind to separate DNA strands. Each hydroxyl group of catalyzing serine residue in all 4 recombinase molecules acts as a nucleophile, and it attacks the substrate DNAs and forms 5' phosphoserine linkages as shown over here, and then ligation and rotation takes place, and then the products are formed. 2 of the 4 recombinase molecules that are covalently linked to different substrate DNAs rotate. You can see here there is a rotation. So, one of these blue has moved up, while the pink color unit has come down. The DNAs rotate with respect to the other pair of the recombinases and religate their attached 5' phosphates to the 3' of the unrotated DNAs. So, this is a little bit different from the earlier case. Here there is no any formation of the Holiday junction. Serine resolvases are site specific recombinases that carry out excision reaction and resolve large fused replicants into smaller separated ones. Some resolvases are encoded by replicative transposons and resolve the transposition product in which the donor and recipient molecules are fused into separate replicants. Other resolvases are encoded by plasmids and function to resolve plasmid dimers into monomers. Both types are therefore involved in the spread and maintenance of antibiotic resistant genes. Serine is a resolvase of the serine recombinase family that is encoded by various Staphylococcus aureus multi-resistance plasmids. What are the biological roles of resolvases? Gamma delta and TN3 resolvases are encoded by replicative transposons. Transposition creates a branched intermediate. This is processed by the host replication and repair machinery to yield a co-integrate in which both the donor and recipient replicants are fused. Finally, resolvase action at a rest site within the transposon resolves the co-integrate into the original donor and the recipient carrying a copy of the transposon. Resolvases like beta and sin are encoded by plasmids. Rescue of a stalled replication fork by a homologous recombination mediate pathway can lead to a Holiday junction behind the rescued fork. In this diagram, you can see homologous recombination of a co-integrate plasmid containing the TN3 transposon. This is an important part of the replication of this transposon, which itself has a role of the spread of antibiotic resistant genes. Let us see the structure of a syn tetramer, and in the reaction pathway there are 2 inactive site 1 which are bound dimers and come together and form a catalytically active tetrameric species. DNA cleavage resulting in phosphoserine intermediates is followed by 180-degree rotation of one rotating dimer relative to the other. Then religation happens yielding the recombinant products. Formation of the tetrameric species can be triggered by a accessory protein bound to regulatory sites on the DNA. Let us study about the biological roles of resolvases. Gamma delta and TN3 resolvases are encoded by replicative transposons. Transposition creates a branched intermediate. This is processed by the host replication and repair machinery to yield a co-integrate in which both the donor and the recipient replicants are fused. Finally, resolvase action takes place at a rest site with the transposon within the transposon resolves the co-integrate into the original donor and recipient carrying a copy of the transposon. So, in this diagram you can see the homologous recombination of a co-integrate plasmid containing TN3 transposon. This is an important part of the replication of this transposon, which itself has a role on the spread of antibiotic resistant genes. Resolvases like beta and syn are encoded by plasmids. Rescue of a stalled replication fork by a homologous recombination mediated pathway can lead to a holiday junction behind the rescued fork. Let us look into the structure of a syn tetramer and the reaction pathway. Here there are two inactive site 1 bound dimers which come together and form a catalytically active tetrameric species. So, this is inactive and these are also inactive. When they come together, they form a catalytically active tetrameric species. DNA cleavage resulting in phosphosericin intermediates, this is followed by 180-degree rotation of one rotating dimer relative to the other, and then religation happens and it yields the recombinant products. So, you can see here the cleavage occurring, and then there is a 180-degree rotation. So, these goes up while this comes down over here, and due to this rotation you can see here the differently colored DNA molecule which represents a different DNA molecule is flipped, and due to ligation it is joined with the other DNA molecule. So, this is one DNA molecule denoted by one color, this is another DNA molecule denoted by another color, and here you can get a recombinant product as denoted by the presence of both the colored fragments of DNA and in both the products. Rest sites and topology of the synaptosome. Sericin resolvases has rest sites. Specific recognition sequences around 12 base pair each. For individual resolvase, subunits are shown as colored boxes. This green is for the crossover site, always an inverted repeat, purple for accessory site that form direct repeats, and yellow for accessory sites that form inverted repeats. The recombinase dimers bound to the accessory sites are catalytically inactive, and these sites always differ from the crossover site in the length of their central spacers and relative orientation of their half sites. So, site 1 is basically the crossover site and site 2 is the accessory or the regulatory site, and there is a site 3 as well at the end of site 2. So, there are various examples for this particular pattern. You have syn and beta. For the second type you have TN552, then for the third type you have PAR-A and then T2501, and the fourth type you have TN3, gamma delta and TN21. The gamma delta rest site. So, you have site 1, site 2 and site 3 as shown earlier. The 3 binding sites for resolved dimers are indicated by 1, 2 and 3 with the open arrows representing the 12 base pair resolvase recognition sequence. This L and R indicate the left and right halves of the 3 sites. So, for first site LNR, second site also LNR and third also LNR. The vertical arrows So the crossover point at site 1 here. The distance between the centers of each binding site and the length of the intrasite spaces are also shown in this figure, around 53 base pairs here and 34.5 base pairs. What are the various application of site specific recombinases? Due to the simplicity in mechanism of deletion, insertion and inversion of the DNA sequences, different site specific recombinases have been applied to in vivo genome engineering, and in some of them it has been used successfully to mediate knock-in and knockout. We will be discussing about the use of some of these site specific recombinases in our later lectures for producing knock-in and knockout organisms. And in a wide variety of heterologous genomes, including those from bacteria to higher organisms. Thank you for your patient hearing.